Okay, so we talked about the purines and the pyrimidines. It is important that you know the differences between them. Purines bind with pyrimidines, and do know what they look like structurally. And of course, you've been provided with a handout for that. Now we're going to get into DNA replication, and I was thinking that maybe I had a slide, yes, that's going to kind of help us with this. So let me clear these annotations, and we'll just begin from the beginning. All right, so we've established that DNA has to form this attachment to its bases. And remember, according to Chargaff's rule, he said that you're going to have everything to a 1-to-1 ratio. So for every cytosine, there'll be a guanine. For every adenine, there'll be a thymine. And of course, we're going to learn later that that's not always the case, and some freaky stuff really happens. Okay, so let's just pretend like this is our DNA molecule. And on your paper, you have one that's titled DNA replication activity. So DNA replication activity. And it's got some directions, and it says something like use the image below and the DNA cutouts to help the DNA molecule replicate itself. And okay, so let's make a connection to this. Why replicate? So why are we replicating? Well, if you guys remember, in the cell cycle, we had a couple of situations. So we had S phase. Okay, I'm not sure what that's supposed to be. We have G1, and then of course we had G2, and then you had your mitosis and cytokinesis. And this was also the same for meiosis. So looking at the S phase, DNA is going to replicate itself, and this is what it's going to look like. Now, when you're finished, identify the original strands and the new strands. So I would think about color when you did this. I would color your strands or whatever, just so that you could tell them apart. Maybe label them if you want to, old strand versus new strand, or create some kind of key. And there are little tiny cutouts, so good luck with that, some of you that like to lose your parts. So, but let's replicate this molecule. So let's say we've got this beautiful enzyme, and her name is helicase. And helicase is supposed to unzip and unwind the DNA molecule. And what that's doing is it's creating a template. Let me erase that and make that better. No room for error here. All right, so we got A, C, G, T, C, G, A on one side, and of course your other template is so on and so forth. So you guys following Chargaff's rule, you're going to make 2 strands of DNA. And so I want you to mash pause, and at this point I want you to kind of construct your or demonstrate this really basic concept of DNA replication. So hit pause now. Okay, so we looked at a really generic form of replication, and you guys did all of your little cutouts, and you did all of your coloring, and so you're really trying to tell the difference between what your old strand is versus your new strand. And, you know, as we said, this is when DNA is cut by the helicase, it's really just creating a template for a copy. I mean, this is, it's a beautiful mechanism for DNA to fix itself. And I do have a link that's going to, help us with the next, because I think this is your last set of worksheets. And I hate calling them that, worksheets, or illustrations that kind of go into DNA replication. So let's take a look at that now. I just kind of want to make sure that this is working. So, so of course, whenever you guys are ready, you can just click, play, and you can listen to this.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex.
Okay, and I'm going to pause it there, and I'm going to kind of pull your attention to the DNA replication, the sheet that I have. You know, she was talking about a replication fork. So if you can, circle the fork or color code it as it's listed at the bottom of the page, because I think it says circle the replication fork in brown. And of course, you saw the DNA helicase that I had mentioned earlier. But let's continue listening.
Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step.
Okay, so I'm going to again pause this and kind of draw your attention to, she said there's a strand coming out at the top, and this is called our leading, and then you have a strand at the bottom, and it's called a lagging. And the replication mechanism for these two strands is quite different. So make sure that you listen and pay attention as she highlights those areas.
The DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation.
So on your paper, down there at the bottom where it says lagging strand, I want you to make a note of Okazaki fragments. I'm not going to ask you to replicate the lagging strand, because as she kind of introduced us, this is way more complex and it kind of runs backwards than the leading strand. I really need you to just be able to identify and understand the different parts and mechanisms that are involved in replication. So let's continue kind of making note of different areas on our handout. She did mention DNA polymerase, and again, this is going to be an enzyme that is going to attach the nucleotides that you had learned about earlier to the DNA template. So let's listen.
These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
Okay, so she's given us quite a bit of stuff to think about in terms of DNA, and as she said, she had left out quite a bit of information just to keep from confusing us. So, okay, so now as we've seen, we noticed that DNA is quite complex and her mechanisms are quite involved. But with this last part, I really want you to take some time and I want you to color code all the different parts, color code all your bases, your template strands, circle the replication fork, identify what your enzymes are. It is so important that you understand DNA replication and the importance of this, and just, I don't know, gain an appreciation for just how beautiful. Now, associated with that whole activity, of course, there's a DNA replication sheet that's asking you questions about the functions of various enzymes and the steps involved in DNA replication. And of course, it gives you some fill in the blanks kind of thing. So if you want to go ahead and try to answer those, that's fine. And then this will give us something to kind of work from on Wednesday.